Hi, this is Dr. Dan Guerra coming to you from VerEvMed Studios in conjunction with Authentic Biochemistry Podcast. I'm coming to you from the Inland Pacific Northwest on a beautiful Tuesday late morning. It's the 13th of November, 2019. Today, as you can see from my PowerPoint initial slide here, we're going to continue on part 2 of T lymphocyte molecular and cellular Diaevenontology. So last time I introduced once again, because I've done it several times in lecture, what I mean by Diaevenontology. It's a new paradigm for understanding the interaction of individual biological organisms in their environment and how the environment, along with genetics within that system that is an individual organism, interact in such a way that a neuroimmunoepigenetic file is generated because of changes in gene expression in such a way that it tailors, modifies according to experience, that is empirical understanding of the world around you, in such a way that the body responds and continues to respond either episodically, depending on, let's say, the invasion of a pathogen or change in temperature, all the way down through years and years and years, especially when you think about processes such as learning. So the epistemological association here is also important because this is, of course, a biochemistry, molecular genetics lecture, and I want you to learn something. But the important point is that Diaevenontology is a paradigm that makes a trigonal planar association with how genetics and environment interact with this third component, which is basically how there is a arc or there is an axis that connects the two. And that axis, again, has to do with epigenetics and has to do with a neurological response that is formulated and that is expressed and organized around what used to be called the classical immune system, both innate and acquired immunity. That's why I'm using T lymphocyte biology today to explore the Diaevenontology. Hopefully at the end of these lectures, there'll probably be 2 or 3 more, we'll get to having some kind of consensus understanding that we can generate a dialectic and follow through perhaps with a logical argument that will get us a little bit further down the road to what I'm trying to get at here. So let's just get started with the lecture. Okay. Again, I'm Dr. Daniel J. Guerra. That is my email address down there, Facebook, and these are the 2 links that you can go to for all the podcasts down here. And the second link down here is my where Apple connects with Authentic Biochemistry Podcast. Of course, there are 6 or 7, maybe more than other platforms you can acquire me at on my Authentic Biochemistry Podcast. And also there is the YouTube channel right there, which will get you to all the video lectures from Authentic Biochemistry and VerEvMed. Those are the 2 logos for the companies I represent, co-own, own this one outright. And let's just get started. Again, there's a date, and we're going to talk about T lymphocytes. So let's go. All right. I want to step up here a little bit, and I don't know why we're getting a slow response, but we'll get to it. Here we go. How are T effector cells generated and regulated? This comes from a paper published in Experimental Molecular Medicine just recently. You can see that it was published in 2019, only probably a few months ago. What does it tell us? I guess I don't need these for reading this. CD4 T cells, which are kind of the progenitor cells, play key roles in acquired lymphoid immunity by orchestrating cytokinogenesis, that is the synthesis of new T lymphocytes, while at the same time differentiating to the mature cellular lineages, including these lineages, which we will cover in some detail, Th1, that's T helper cell 1, Th2, Th17, important for viral infections, and Tregs, which control or suppress normally the pro-immune response generated by Th1, 2, and 17. There are a couple of other T cells we're going to talk about today, which normally are not brought into discussions in primary immunological lectures, but we're going to, of course, cover them here because we're a graduate level discussion. We're also going to talk about T cell receptor activation here, right? Because all of that is associated with the different lineages. So each CD4 lineage expresses a suite of unique transcription factors which control gene expression at the genetic level, and that drives their terminal differentiation. And those include but aren't limited to these transcription factors. Tbet as transcription factor for Th1 cells that can go on to clear intracellular pathogens. And it does so by inducing transcription of the interferon gamma gene. Okay? Similarly, Th2 lineage express interleukin 4, 5, and 13. Those are, of course, pro-inflammatory cytokines. And in response to helminths, right? So now we're talking about parasitic infections. And they use the transcription factor GATA3, while Th17 cells express a unique transcription factor called ROR gamma T. More on that later. And that is a lineage determining factor, and that produces cytokines, because this is Th17 cells, interleukin 17A, 17F, and then interleukin 22, all of which can be inflammatory and help associate Th17 cells with infection. Those are all regimented to eliminate extracellular bacteria and fungi. But there are dark sides to T cell activation, obviously. That includes a Th17 autoimmune mediated multiple sclerosis, which is a major disease in humans, and rheumatoid arthritis, which is also an important high morbidity disease. And to control that overreach by those cells, by the acquired immune lymphoid pathway, that's what I mean by those cells, another adaptive member of the T cell lineage that blocks, not blocky, that's cute, the pro-inflammatory drive are the Tregs. So they T-regulate. That means they're going to suppress, as I said before, the Th lineages. Tregs tank differentiation and proliferation of effector T cells. Effector T cells are those Th cells. And they downregulate, because of that, inflammation, thus checking autoimmunity as well as many potential allergic responses. Okay, so what else we want to say here? Here's a paper that was published in 2017. Sorry, my mouse isn't working well today. The Tbet transcription factor is an essential regulator of effector differentiation, as I've said, and it functions in multiple different immune lineages. That includes the progenitor cell CD4s, CD8s, the natural killer T lymphocytes, the ILCs, which include a natural killer lineage, and even B cells and plasma cells. Okay, so that transcription factor is wide-ranging of activity. So inflammatory cytokines in general, which can be generated, for example, by epithelial cells, can induce Tbet transcription in a graded manner, which in turn regulates distinct differentiation outcomes. Okay, so the level or the concentration of Tbet and where it's expressed in those different T lineages is going to then manage and organize the T lymphocyte overall lineage to deal with a specific invading system, right? Because remember, this is still immunity. So Tbet can act as a fulcrum between Th1 and TFH cells. Okay, those are follicular helper cells. That's at their level of differentiation, of course. Also pathogenic and non-pathogenic Th17 cells, and CD8 effector and memory T cells as well. So the whole lineage can be graded and regulated when you start with Tbet as a global transcription factor. So during infection, for example, Tbet induces a chemokine receptor called CXCR3 that happens on multiple different lymphocytes. So now you're able to express this receptor, and you're able to signal and alter the entire expression of a subsequent downstream distal association of gene expression, because now you have this chemokine receptor on the surface. And that gives you multiple different lymphocytic interactions. That promotes an effective immune response by allowing co-colonization or co-localization, of multiple cell types within the appropriate tissues for the targeted effector function. You can see how quickly this expands, like a geometric function, right? Like a binomial expansion. It's expanding. And each of these cell lineages is going to give you a different repertoire of biological activity, right? All still regulated at some level by effector molecules such as cytokines and chemokines working in tandem to control the expression of transcription factors, the graded level of those transcription factors, and then that regulates how much chromatin remodeling occurs, and then what genes are expressed and how much of those genes are expressed at the transcriptional level. Okay. You see that this quickly becomes a beautifully orchestrated system, right? A symphony of T cells, and here they are, right? So there's your CD8 progenitor cell and CD4. CD8 cells, when you increase T-BET transcription levels in the presence of interleukin 12, will make a T-BET blimp type cell, and that's going to turn into a cytotoxic T cell because now it's expressing that chemokine receptor. That's what's supposed to be up there. So you CXCR3. For example, that's what happens to CD8 cells. What about CD4 cells progenitor? In the presence again of interleukin 12, okay, you can then make high levels of T-BET. You still get that chemokine receptor there, but you don't have this other transcription factor functioning. So that's going to be the Th1 lineage. Now, in the presence of interferon gamma, either the cytotoxic T cells or the Th1 cells can then ultimately cause Th1-induced inflammatory responses. And then Th1 inflammation is going to include the expression and production at the translational level of these proteins. The CXCL9, that's going to be a chemokine, not the receptor. Chemokine 10, the receptor again, make more of this receptor for these cell lineages. So there's going to be those are that receptor's ligands, those two chemokines. So that's going to give you full-blown Th1 pro-inflammatory system. Okay. So CD4 cells in the presence of interleukin 12 and 6 are going to give you this new transcription factor here. And along with T-BET at a lower concentration, you're supposed to see it a smaller amount. And that's going to give you these T follicular cells. So the T follicular helper cells, shown right here, are a specialized subset of CD4 T lymphocytes that are first identified actually in the human tonsil, which, years ago when people used to get a lot of sore throats, they'd get tonsillectomies. Well, that would remove your production of T follicular helper cells. So that's probably something that wasn't invade as an understood byproduct of the removal of tonsils at the times. At any rate, that's when they were first identified, and they play a critical role. By the way, you also make T follicular cells besides in the tonsils. I should say that. Of course, I think that's obvious. But they play a critical role in protective immunity because they help B cells produce antibody or immunoglobulins against all kinds of different foreign pathogens and epitopes associated therein. So T follicular helper cells are located in secondary lymphoid organs, such as the tonsils, and that includes, as it says here, the tonsil spleen and also many, many lymph nodes. And there is the link for that if you want to read more about it. All right, so that's what's going on with these T follicular helper cells. Next, okay, you have a B cell, right here. It's going to be in the presence of a CD4 T cell lineage expressing these transcription factors, and then in the presence of interleukin 21, interferon gamma, are going to turn into this BCL6 moiety of cell lineages of the B cells, and they're going to start producing an IgG2A system, right? So you're going to start making immunoglobulins. Those will eventually turn into plasma cells, where they'll make a lot of this BLIMP1 transcription factor. Still T-BET, because I told you T-BET is important for all of these lymphocytes, not just T lymphocytes. There's a B cell, for example. And that's going to start generating IgG2A, and that's going to go into the serum, okay? So that's how B cells are regulated by T cell modification of gene expression. going directly from the progenitor cells to these ones expressing these two transcription factors in the presence of those cytokines, pro-inflammatory cytokines. There's Th17 cells. They're going to be expressing this transcription factor called ROR gamma T, which is more about that later. And in the presence of the cytokine interleukin 12, excuse me, let me get out of the way here, are going to express now more T-bet. They're going to pick up this chemokine receptor, and then they're going to go directly into enhancing the Th1-induced programmed inflammatory response. So you see how T cells interact with one another to produce an inflammatory response. Now here's the ticket out of this race towards inflammation. And there's your Treg cells. Treg cells have a unique transcription factor. Actually, Th17s also express it sometimes, and they cause them to differentiate differently. But the important, if you want to think about what's the transcription factor that goes with Tregs, what's the one you always see? FOXP3, okay? So that could be on an exam, for example. So FOXP3, in the presence of interleukin 27, starts expressing the T-bet transcription factor 2, as well, I should say. So you've got FOXP3, T-bet transcription factors, you've got the chemokine receptor, and that, when you make that, you make a T-regulatory cell. That's going to suppress all this inflammation. See, all that Th1-induced inflammation is going to tank. That's what this green is supposed to show. It's going to slow it down. So we need T-regulatory cells because they're going to control the inflammation response so it doesn't overdo that induction. And then once that is overdone, the differentiation and the proliferation of those cells can lead not only to a chronic inflammatory system, such as in type 2 diabetes and in various prodromal stages of cancers and cardiovascular diseases, for example, because of the synthesis of a lot of reactive oxygen and DNA mutations, kinds of things that are hallmark paradigmatic for those diseases associated, of course, with aging and vagaries of living. Those Treg cells are there always, that lineage is always there to blunt that response, in fact, to shut it down. Not completely, but to keep it suppressed sufficiently so you don't eventually lead to something like an autoimmune disease, such as lupus, systemic lupus. Okay, so this comes from Trends in Immunology, and you can see there the citation. All right. All right, now, next slide. I will be one moment, be right back. All right, that makes it so that I'm not a big white thing on the screen. My producer director isn't here today, so I had to change the lighting in the room, which basically meant closing down the blinds that generate all the sunlight from the western vantage point of my studio. I'm in the upstairs studio right now, actually. Okay, so we're done with this talking about that. Let's get us moved out of the way here. I don't want to get rid of me entirely because that looks weird. Okay, Treg regulation, experimental molecular medicine. Again, this is the 2019 paper. So the transcription factor FOXP3 is an X chromosome encoded Treg cell lineage determining factor. Okay, X chromosome. Isn't that interesting? Cytokines TGF beta and interleukin 2 induce expression of the FOXP3. Now, I'm pointing out again about this X chromosome encoded Treg lineage determining factor because I want you to understand there is some sexual dimorphism when it comes to inflammation and diseases that occur in either men or women. Okay, that's correct because of gene dosage effects. You know what I'm talking about. So interleukin 2 induces this paradigmatic pathway called JAK-STAT, which is basically a kinase cascade, and it initiates ultimately FOXP3 transcription. When TGF beta, that's of course a pro-inflammatory cytokine, binds to its receptor, SMAD2/3, this is going to be a co-adapter intracellular molecule, undergoes phosphorylation, and what it does when it gets phosphorylated, it translocates to the nucleus. Phosphorylated SMAD2 dissociates, binds to a conserved enhancer region in the DNA, or a conserved non-coding sequence, those are called CNSs, at the FOXP3 locus, right? This is all at the chromatin remodeling stage within the DNA, right? The chromatin. And what happens there, of course, is that you get unique binding domains for those specific transcription factors that follow distally, right? So you're going to have more than one transcription factor. Remember that this FOXP3 is just kind of the global initiator of the process. So one of these conserved non-coding sequences, which is a component of the enhancer region of the DNA that's going to code downstream for transcripts that are going to make this cell lineage a Treg lineage, right? Yes, of course. CNS1 is actually unnecessary for thymus-derived Tregs. Those are the progenitor Tregs. So it's not necessary for generation of them. And that makes sense, right? Because you're going to do all this phosphorylation cascade, kinase cascade, and subsequent transcriptional regulation. You don't want to have the thymus having to regulate and officiate an interaction of some kind of induction associated with TGF, because what you want to do is have that programmed at a different level. So you don't want to use CNS1. So this, again, shows you more of the complication, but also how it's absolutely beautiful in the way that it's regulated in terms of how you make Treg cells. So I hope you understand that. I'm following along. I'm sure you will. Now, CNS1 plays a prominent role in all the peripheral Tregs. Those are the ones that are in circulation, right, or resident in tissues or associated with tissues. So that tells you about CNS1. CNS2, okay, that's a conserved non-coding sequence, all associated with the enhancer region for the FOXP3. Has a binding site, of course, and it contributes to Treg cell stability, while CNS3, with its CREL binding site, for those of you that are oncologists, I know you've heard of CREL before, because mutations in that can induce certain kinds of tumors. CNS3 has a CREL binding site, and that actually increases T regulatory cell generation, that is, division. Right? All right. And also ultimate differentiation with CD4s. So, yeah. So FOXP3 activates Treg signature genes, including CTLA4, interleukin 2RA, tumor necrosis factor receptor F18. That's a superfamily member 18. It's called a superfamily because you find that molecular signature in a lot of other receptors. And the ICOS, which is the inducible T cell co-stimulator. All of those participate in the suppressive function and ultimately the differentiation. In this case, not necessarily terminal differentiation of the Tregs. More on that later. So demonstrate the physiological consequence of FOXP3 mutation or deletion. This results in lymphoproliferative diseases, including autoimmune diseases, and is characterized ultimately by multi-organ lymphocyte infiltration. This can cause high level of morbidity and actually mortality once the inflammatory response gets totally out of hand like that. All right. So now let's get into more detail here. Publication from 2016 in Cell Reports. Deficiency in either ID2 or ID3 results in loss of distinct CD8 effector in memory population cells. This is from Nature Immunology way back in 2011. Always important to go back and look at the scientific literature to determine how you got to 2019 paper. And you don't do it just by using the references in the 2019 paper. You do it by looking at the overall process. This is where Diaevenontological paradigmatic understanding comes into play. You look at the overall process, and yeah, you can use that to find search terms for papers, but you need to go right to PubMed. You don't want to ever use Google, and you also don't want to get so narrow that you're using search terms that you miss something. You want to have a pretty broad net, and then because you know a lot about the system because you've been studying it, you know what papers can help you formulate an overall understanding of what's going on in T lymphocytes, right? All right, so that's the way I do it, right? So with homeostasis with wild-type Treg cells, ID3 then is going to block this pathway. E47, which is going to turn SPI-B on, which would normally turn on SOX-S3, which normally then would tune down that would tune down FOXP3. But in a wild-type Treg cell, you have plenty of FOXP3, so you'll be able to block the inflammation caused by all the effector T cell lineages. Now, in ID3 minus cell lineages, this is showing you yet another factor, okay? Another factor that's a protein that's going to be associated with altering a dysfunctional regulation of Tregs, right? You get now, because you have this mutation, or you've lost ID3, you don't have it. Now, what happens is because you can't block this, E47 turns on. That turns on SPI-B. That turns on SOXS3. And what does that do? It tanks FOXP3. So now you got basically FOXP3 deficiency. So you are really lousy Treg lineage because you got FOXP3 downregulation, and now your effector T cells can go on to induce inflammatory responses. See how that works? So that says this down here: regulatory T cells suppress the development of inflammatory disease, but our knowledge of transcriptional regulators to control the function remains incomplete. Here they show the expression of this ID2 and 3 in Tregs is required to suppress development of inflammatory disease. They found T cell antigen receptor driven signaling initially decreased the abundance of ID3, okay, which then leads to activation of the follicular regulatory T cells. I told you we're going to talk about them again. And that's going to turn on a specific transcription signature. And then, however, there's a sustained lower abundance of ID2 and ID3 interfered then because of that with a proper development of the TFR cells, okay? Depletion of ID2 and ID3 expression in Treg cells then results in a compromised maintenance and localization of the Treg cell population. Thus, ID2 and ID3 enforce the TFR cell checkpoints, okay? That's the T follicular receptors checkpoints, and that controls maintenance and also homing of the Treg cells. So that's how that whole process works. So, again, that all customer cell reports paper published way back in 2016. Some of the other things I got from ID2 and ID3, the progenitor to that paper was published back in Nature Immunology in 2011. I want you to look at all of those references because I want you to look them up, and I want you to get the whole global understanding of how I see the regulation of T lymphocytes, because you can see it the same way, of course. Okay, here's a paper published in Cell way back in 2011, talking about now transcription factor interleukin 6, hypoxia, reduction in molecular oxygen, increase in reactive oxygen, increase in nitric oxide, fumarate or succinate. Those are what? Tricarboxylic acid intermediates, right? Krebs cycle intermediates. Oh my gosh, how's that work? Every time you think of any cell lineage, I want you to just take this and keep it well controlled and up in front and center in your prefrontal cortex. Anytime you're talking about any cell lineage, right now we're talking about immune cells, which are going to regulate lots of other processes in the system, right? I want you to understand that each of those cells have a bioenergetic requirement. And that bioenergetic requirement is going to involve whether or not they're using fatty acids, glucose, or amino acids for their ATP synthesis. And when you're talking ATP synthesis, you're talking NADH production, nicotinamide adenine dinucleotide reduced form. And one of the ways you make NADH is via the dehydrogenases in the TCA cycle. After the production of acetyl-CoA and oxaloacetic acid from pyruvate, if you're coming from glucose, subsequent to glycolysis, right, making pyruvic acid, or if you're running it through beta oxidation of fatty acids, where you're generating FADH2 and NADH directly into the mitosol, which means you're going to block the dehydrogenase in the TCA cycle, and you might pick up intermediates that are distal to those dehydrogenases, such as fumarate and succinate. Now, if you remember your biochemistry, I know you follow along. If you don't remember it, that's okay. That's what I'm here for. But that's the complexity of this, right? It's a beautiful complexity. All right, so let's take a look at this. Move this box over. My mouse just isn't working today, sorry. All right, so when you have interleukin 6 and hypoxia up here, you're going to produce hypoxia factor, this HIF1, okay? And so what's that going to do? In Treg cells, it's going to interact with FOXP3, okay, this protein will, and then it's going to generate this entire complex of proteins, PVH, LE3, FOXP3, in association with the HIF1, and then it's going to become ubiquitinated. When that happens, because of HIF1 generation, okay, the hypoxia-inducible factor, because it was induced by hypoxia, low oxygen tensions in the presence of those interleukin-6, which again is a cytokine, or high reactive oxygen, high nitric oxide, and those two distal to the dehydrogenases, TCA cycle intermediates, right, fumarate succinate. You're going to just start degrading FOXP3, and you're going to have a prevention of Treg differentiation, okay? So let's go down here and say nuclear receptor that binds DNA as a monomer to the retinoic acid response elements, that's the ROR, contains a single core motif half site, and there it is, that's the actual DNA, right? 5' AGG TCA 3', that little piece of that little 6 nucleotide fragment that's going to be a component of the ROR element in the DNA that this complex is going to bind to and then allow for all those proteins to be, particularly FOXP3, to be degraded, okay? All right, so all of that's going to happen. Key regulator of cellular differentiation, immunity, peripheral circadian rhythm, as well as lipid, steroid, that's of course another lipid, xenobiotic and glucose metabolism. That all comes from a PubMed, several PubMed papers. You can take a look at them. Considered to have intrinsic transcriptional activity, they have some natural ligands like oxysterols, which are generated from cholesterol becoming oxygenated, and they act as agonists. For example, 25-hydroxycholesterol is one of them. Or inverse agonists, it's like 7 oxygenated sterols, different than 25-hydroxycholesterol. And they enhance or repress the transcriptional activity respectively. Recruits distinct combinations of cofactors, that's showing here, and they target gene regulatory regions to modulate transcriptional expression depending on the tissue, time, and promoter context. You see there's all of the members of the orchestra, the molecular orchestra involved here. So Treg cells, I told you what happened in Th17 cells. The hypoxia-inducible factor then is going to produce this nuclear receptor DNA. It's going to generate the ROR gamma T, retinoic acid response gamma T transcription factor. You're also going to, what that's going to cause is the production, because these are card-carrying Th17 cells, pro-inflammatory cells. They're going to make a lot of interleukin-17, okay? So now you see, and you can regulate differentially Treg, you're suppressing the suppressor cells, the Tregs. At the same time, you're tuning up Th17 cells, okay? Excuse me again 1 minute. Sorry about that. My dog, Rocky, the partially the producer of the show, was playing around a little bit in the studio, so I had to put him outside. So sorry about that. All right. So you get the idea. You have differential expression of these different T cell lineages that can be induced from on above, right? So you get hypoxia and you get pro-inflammatory IL-6 production from other cell lineages, including endothelial cells, epithelial cells, right? Organ type cells, right? Vasculature cells. Once you get the hypoxia up, you get hypoxia-inducible factor, and you can totally alter how T cell differentiation plays out. Right? This is how stress from the environment can directly affect T cell modification. So you get the idea. If you get less oxygen tension, right, and there are various diseases which can cause respiratory failure or respiratory suppression. So you're going to get some local hypoxia. What's going to happen is you're going to get like a pneumonia. You're going to get Treg suppression, and you're going to get Th17 elevation. What's that going to cause? A pro-inflammatory response, which can be indicative of certain diseases. You see how terribly beautiful this is. Terrible because it can lead to consequences of morbidity and mortality in the diseased patient. But also, it's beautiful in the sense that the regulation is at the level of multiple forms of proteins that are poised to be induced or suppressed at the level of transcription as related to real-time interactions with the environment. And that's why I talk about how the immune response sculpts and carves the overall cellular phenotypic changes, which are also epigenetically constantly being reprogrammed, some of which that reprogramming, methylation, acetylation, which we'll get to later, can actually be delivered to further cell lineages by normal cell division, methylomes, acetylomes on histones, for example, which will then, with a periodicity associated with that cell lineage and further induction of that cell lineage, either maintain a certain epigenomic alteration of gene expression or allow that epigenomic gene expression to fade out, disappear, right? Like at the end of a movement in a symphony, like the adagio. And then they can be amplified again from those same T cell lineages when you again become hypoxic, right? But now those cells could be in different states of quiescence. There could be different memory cell lineages. There could indeed be different populations of Th1, Th2, and Th17 or CD4, CD8 lineages or B cells, all of which will then alter all the downstream effect of now changing that environment again, retailoring with epigenetic processes such as acetylation of histones. And by the way, those epigenetic changes are going to require the acetyl transfer group, acetyl-CoA. In order for you to acetylate a histone, you have to have acetyl-CoA where? In the nucleus. How are you going to get acetyl-CoA in the nucleus? It's a CoA thioester. Does it stream through the nuclear envelope? Or does it get synthesized there? Is there some alteration of, say, fatty acids in the nucleus that allow for partial oxidation of those fatty acids even in the nucleosomal regions? These are unknowns, of course, right now. But what I'm trying to tell you is you need acetyl-CoA to acetylate histones, which can regulate epigenome, which can then alter gene expression. And you also need what? Acetylmethionine coming from folic acid metabolism. Also in the nucleus, you could have a methyl donor. Acetylmethionine is a methyl donor. But you also, of course, need the entire methionine pathway, which includes homocysteine, which is another completely embedded regulatory response that is even associated with amino acid utilization in the TCA cycle. So ultimately, this is indeed linked to bioenergetics, always. Okay. So that's not all on this slide. That's just what I bring to the table to explain to you how this is always not working in an isolated system. So whenever you generate pharmaceuticals and you think you're targeting, say, the HIF1 or you're targeting the FOXP3 or any of these proteins you see here, you think, oh, aha, if we can alter the expression of those proteins or we can alter their mobility into the nucleus or we can bind them and then pull them out of creating something like this complex that will become ubiquitinated and go through the proteasomal degradation and get rid of FOXP3. If we think we can prevent that, therefore keeping Tregs up and not allowing for Th17 overproduction and over autoinflammation. Wait a minute, all these other systems are also working, and they're not working in tandem. They're working either in a stochastic or actually a pseudo-stochastic, pseudo-random way. Apparently random, but not random as far as the cell is concerned, right? Because it's responding constantly to the vagaries of the environment by utilizing the immune system. So these cells themselves will then become the agents of alteration, altering epigenetic signatures in other cell types and other vascular beds, for example, in either inhibiting or sequestering and altering disease progression, such as metastasis, or enhancing the production of tumor cells, tumorigenesis, or having any effect on tumorigenesis at all until later in the life cycle of the organism when there's more reactive oxygen produced, you see, as aging and senescence is known to be associated with. Or because of undue exercise, you can make more reactive oxygen. And you can also become hypoxic with a lot of excessive exercise. You see how this works? All right. This comes from a paper in Frontiers in Immunology published, 4 years ago, a little bit more than 4 years ago. Thymus is producing CD4, CD8 progenitor cells. Then there's a cortical epithelial cell, which is going to be associated with, for example, matrix compatibility complex, which is a self-antigen associated system with a low TCR, that is the T cell receptor expression, self-reactivity. You're going to then go to the periphery with that cell lineage because it's interacted with. So this is the classroom stage of the CD4 lineage. This is when they're exposed to self-antigens, and the self-antigens will prevent these cells from recognizing host cells when they get into the periphery. You see what's going on here? That's what this is. That's what this means here. Self-antigen is being early enabling these CD4, CD8 lineages, right? So that they have learned in the thymus to recognize host cells and not become autoimmune. At any rate, when that happens, you have low T cell receptor self-reactivity. You make a CD4 plus naive T cell, the presence of now the expression of the T cell receptor, TGF beta, interleukin 2. You make a CD4 positive, CD25 positive. Those are called co-positive CD4, CD25, right? But you're also expressing that FOXP3 because you're expressing these receptors, and you're altering now the transcriptional repertoire, right? Now you have a FOXP3 positive T cell, and in the presence of TGF beta, you have an uncommitted CD4, CD25 FOXP3 T cell, okay? So Treg cells develop in the thymus and in the periphery. So I'm going to tell you about the thymus in a minute. In the thymus, now we go, CD4, CD8 T cells undergo negative selection. That's what that's called. That's the learning process. That's teaching these progenitor T cells do not go in the periphery and start attacking host cells, right? That's what that means, negative selection. It's going to kill all the cells. Thymus is hopefully never going to allow them to be differentiated. And if they are differentiated, not allow them to proliferate, they'll either go into anergy or they will be destroyed themselves because of apoptosis, right? Okay, so that's what I mean by negative selection. Undergo negative selection and become mature Treg cells. That's thymus Treg cells. So as we go here, so here you have an intermediate self-reactive TGF beta signaling going on, high TCR response, T cell receptor response. Immediately you're going to apoptose. You're going to kill that cell because you jacked up, you amped up the T cell receptor reactivity. So that means that T cell was a poor student in the classroom that's teaching T cells, don't destroy yourself. Don't go out in the periphery and destroy host cells, right? But if you have intermediate self-reactivity, okay, of the T cell receptor, you can now generate a CD4 positive, CD25 positive, FOXP3 minus T cell. Okay, you still don't have FOXP3, so you're not in this lineage now, but now in the expression of interleukin 2 or interleukin 15 or in the ratio combinatorial processes, now you make a CD4, CD25 positive, FOXP3 positive because of these cytokines. See, these cytokines are doing more. That's why I want to teach you about the immune response. The immune response is not just about defense. The immune response is acting as the agency to generate all of the cell lineages that are going to end up generating the phenotype, including the immune defense phenotype, but also the phenotype of all other cells that react with them because of co-epitomic epitope exchange processes on cell surfaces of cell lineages, including epithelial and endothelial and other plasma cells and other lymphatic cells, that are going to alter overall the programming of those cells that are going to recognize this is self, leave it alone, and this is your entire repertoire of T cell lineages that are going to go out and preach the word of what those T cell lineages are going to do for the rest of the organism's life. Right? You're always going to be, so you're stuck with these T cells once you've gone through this negative selection. So, and then you end up just with proliferation, anergy, or what? Apoptosis. You can also get all kinds of alterations in these T cells, like autophagy, which means basically the T cells hang around long enough to alter what? Bioenergetic quotients. So these are always happening in real time. That's why epigenetics and what I call a dialectical event is important. These aren't substances. These are dynamic systems. This poor, sad figure I got from this otherwise great paper, Frontiers in Immunology 2015, is showing you something static. It's not static. This is in dynamic mode, right? Now, the thymus ends up making these T cell lineages, but you see it early on. So it's a developmental sequeon, I call it. And then from that early on process of negative selection, you end up with all these other T lymphocyte lineages. Now, how do you go from this cell to ultimately a committed CD4 positive, CD25 positive, still in the thymus, because these are thymus Treg development going on here. We don't really know that, but we do. But since these last 4 years, we think it has a lot to do with, again, the expression of that FOXP3 and then co-transcription factors, which will be revealed as we go on with this lecture. Okay, so you get the idea. It's an elegant, completely complex process. It only becomes complicated. Complex and complicated, two different things. Complicated means it is difficult to arrange the puzzle pieces so you understand how A goes to B, or if A, then B, or if A, then B, but not B, then not A, right? And you use the logistics of understanding these interactions before you ever set out to think about a pharmacological inveighing to alter those pathways. Because when you're way distal to these processes, you don't know these processes, ultimately, what can you induce? You can induce more morbidity or damage to the system, particularly if you're using prophylactic drugs, right, earlier on, such as immunizations. All right. So I just want you to get that whole idea in your head. So more about this. Lymphoid cells are complex and can interconvert. Here's a paper published on my birthday in 2015. All right. Now let's read this. I know it's a lot, but we're going to stop here. So the adaptive immune system probably arose late in evolution because we don't see it in invertebrates. And it consists of B and T lymphocytes that express recombining antigen-specific, antigen-receptive proteins called antigen-specific receptors. You have naive T and B cells, which we've just been looking at. They're activated by their cognate antigen in secondary lymphoid organs, which we just mentioned to you, like the spleen and the tonsils, right? And they undergo significant cell division, right? You make more of them, and then they differentiate, and they exert their effector function subsequent to all of those processes. In contrast, innate lymphocytes display rapid effector functions. So you have innate lymphocytes. So normally you think lymphocytes as acquired immune response. Sorry, take that old-fashioned idea and bracket it off because it's no longer can be considered dogma. That dogma's gone. The beautiful thing about science, things are always changing. You follow the facts and you try to find truths with a small t so you understand the phenomenology of the biology. Then you can do things like describe it like we're doing today in lecture. Okay. All right. For more than 3 decades, since this whole thing was discovered, natural killer cells, I love those cells, NKs, were the only recognized innate lymphocytes, and they're very important, and we've talked about them in lecture. But more recently, additional innate lymphocytes have been discovered and were considered to be a part of the family of effector cells collectively named ILCs, innate lymphoid cells. ILCs have a lymphoid morphology, so they are lymphoid cells. They lack rearranged antigen receptors. Now that makes them unique because that means they're not going to be responding to foreign antigens the way a normal T cell, a bread and butter carb-carrying T lymphocyte is going to. In other words, they're going to be turned on much quicker, right? That's why they're going to end up being cells like NK cells in a way, right? All right. Lack rearranged antigen receptors and are abundantly present at mucosal surfaces. Now, what's going on with the mucosa? That's where the outside meets the inside, right? The mucosa, think of your enteric system. You digest food. Was that food sterile? No way. Are the microorganisms that you also breathe in that become part of the stream of the saliva that ends up being swallowed, goes through the stomach, goes through digestion and whatnot? No way. That's not a sterile system. So that mucosal surface is you're going to need a whole lot of immune surveillance. And you don't want to be monkeying around with T lymphocytes that are going to require all kinds of tailoring and retailoring. You're going to want to have T lymphocytes that are going to be ready to go now, right? Like frontline Roman legion, like go right now. And the way that functions, okay, is you have this innate lymphoid cell lineage. That's what I'm trying to tell you here. All right. So
The expression of lineage-specific transcription factors and discrete cytokine profiles led to the identification of 3 distinct ILCs because they went looking for them. And they have striking parallels with T helper cell subsets. Interesting, because they're going to cause an inflammatory response, right? The main ILC groups are killer ILCs, comprising natural killer cells, the helper-like ILCs, and those include ILCs 1, 2, and 3. Okay. Group 1 ILCs resemble Th1 cells. No surprise there. They include the natural killer cell lineage and other interferon gamma producing innate effectors. And so ILC1s were shown to depend on TBX21. That's the T-BET. Remember that transcription factor way back at the beginning of this lecture, right? Interleukin 7 and interleukin 15. Group 2 innate lymphoid cells are similar to Th2 cells. Thankful, they're called 2. They're 2, right? So the innate, the ILC2s are ROR-A and GATA3. Those are transcription factors. That's not the ROR-gamma T. That's the retinoic acid orphan receptor A lineage of receptors, transcription factors, because they're going to be intracellular receptors, like going right onto DNA, like the GATA3 does. And it'd be dependent on that, right? And interleukin 7 dependent and produce interleukin 5 ultimately and interleukin 13. That's what's shown here. So sorry for the fuzziness of that. I couldn't get a good reproduction of this, but you start off with a global ILC progenitor cell, which are called CILPs. And so you're going to have if you have ID2, you're going to make an NFL3. If you have IL2 minus, or if it's going to be a mutated form, you're going to have a CHILP cell lineage. You're going to make an ILCP lineage. You're going to be expressing ID2. Remember that from the last couple slides. A different transcription factor here. And ultimately, you're going to make all these lineages that I just told you about. You got cytotoxic, innate lymphoid cells, right? Those are the natural killer cells. You can see the transcription factors that are going to be expressed. And there is ROR-gamma T for the NCRs, right? And these are all going to be part of the ILC3 lineage. So I want you to get the idea, okay? I want you to get the idea that this is how, in the periphery now, you have innate lymphoid cell lineages that are going to fire upon potential antigens at mucosal surfaces. And this is really important so you don't get sick, for example, every time you ingest food or you swallow water or something. That's why they're there. That's how they work. All right. So I'm going to finish with this. There are 2 variations of recombinant adaptive immune systems. So they're called AISs. And they arose in vertebrates about 500 million years ago, for those of you that would like to know the evolution of this, which I, of course, do. The jawed vertebrates diversified their repertoire of immunoglobulin domain-based T and B cell antigen receptors, mainly through the rearrangement of those VDJ segments, gene segments, and what's called somatic hypermutation. For those of you that have an immunology course, hopefully you can go back and look at that. Remember what I said, how we teach that. I'm not going to go through that right now. Someday I can do a primer on the entire VD and VDJ recombination mechanisms that lead to B cell lymphocyte lineages. That's a full-blown story. And then we can go back and talk about TCR, T cell receptor, and they also arise from recombination of various components because of things like hypermutation. Okay. Now, AIS, remember that is adaptive immune systems of jawless vertebrates, contains various lymphocyte receptors, those are called VLRs, thank you. And they are generated through recombination of diverse leucine-rich repeats, those are proteins with the LRRs. Transposon-like recombination activation also occurs uniquely, and it's associated with the origin probably of the AIS. We think that's where it came from transposons. Transposons carry with them the machinery to go into chromatin and rearrange it, at the same time have the enzymes expressing so that they can do that rearrangement. They're like, everything goes, it's like a Viking ship, right? The Viking ship has all of the Vikings in it. It also is seaworthy, and it can go from, say, Denmark to Britain and wreak havoc, right, in the 9th century AD. It's like that. That's how I look at it. I know it's silly. It's a silly analogy, but it works, right? So bear with me. Well, of course, because you're a captive audience. Whereas mutational activation induced cytidine deaminase is found in higher vertebrates. So what does cytidine deaminase do? So you have the basic, first let's say the basic adaptive immune system. It features two interactive T and B lymphocyte arms in both jawed and jawless vertebrates, and that arises then from innate immunity. So let's look over here. There's something called activation-induced cytidine deaminase, okay? It's a central regulator of B-cell differentiation. So you have cytosine, you remove the amino group, now you make uracil. So what happens there, that's a deamination, okay? Because you're getting rid of an amino group. You're going to go from a CG nucleotide to a UG, and then when that gets replicated to DNA, you're going to make an AT, right? So now what have you done? You've made a mutation because of cytidine deaminase, that enzyme, which is activated in these innate lymphocyte cell lineages. Yeah, good. So functioning by deaminating cytidine, okay, coupled with base excision or mismatch repair, that's the kind of recombination repair mechanism to deal with that mutation, to deal with that deamination reaction, which is going to give you this intermediate UG, right? And DNA replication is going to try to fix it, but it's not going to make CG, it's going to make AT, right? Totally different nucleotide. Now, depending on where that is, it may have no effect at all, or can have a tremendous effect. For example, if it's an enhancer region, right? Or a cognate promoter region, or maybe be a splice region between nexon and intron, could have a tremendous effect on gene expression. You see how that works. All right. So it functions as a mutator for that reason. Thus, essential an adaptive immunity class switch. This is all about adaptive immunity now, because it associates with recombination, somatic copper mutation in B and T cell lineages, helps generate a diverse and high affinity repertoire of immunoglobulin isotypes. This is part of how you make that entire, 10 to the 11th, 10 to the 12th type of immunoglobulins by this deamination reactions. It's also active in epigenetic signature methylated cytidine. Yeah. So if you're methylating that carbon 5 on a cytosine residue, you're going to alter the epigenetic profile, because now you're turning that cytosine residue into, and because you've turned that into a uracil, because you deaminated it, it's basically the product of the reaction. And then when you do replication, now you're making an AT, you've lost that methylation signature, okay? That can totally reprogram epigenetically an entire lineage of gene expression. So that's amazing, right? So when you couple it with repair, which has to work here, you can get active removal of methylated DNA this way. Now, I got all that from Biomed Journal 2014. All right. So we're going to stop there. This will get us back out at the 100,000 feet above the Earth's surface level and just talk about all these T cell lineages and B cell lineages and how you get there, all the lymphoid cell lineages. And I want to do that, right? I want to get back to that, but not now. So let's close this. All right, let's get rid of that. Put me right there. All right, so what I want to do now is end this second in our T lymphocyte biology, the Diaevenontology of T lymphocyte biology, physiology, molecular genetics, and biochemistry thereof, and say that the next step is to start putting more of an understanding of how all of this complex T cell and B cell innate immune response can be understood when we start looking at diseases, particularly autoimmune diseases, as per requested. So I'm going to do what I always do at the end of these wonderful lectures. I'm going to say, this is Dr. Dan Guerra saying bye for now.